Welcome everyone to the SBA seminars. My name is Zach LeBlanc, and before we get started, I just wanted to say that SBA Brisbane acknowledges the Turrbal and Jagera people as the traditional owners of the land on which this event is taking place, and we pay our respects to their elders past, present, and emerging. We recognize that this land has always been a place of teaching, research, and learning. So I'd just like to welcome anyone who's coming out for the first time to an SBA Brisbane seminar. SBA is a small group that likes to put on events for people that are interested in synthetic biology and, as far as you can stretch that term. And we put on just casual meetups as well as seminars with distinguished speakers like we have today. And if you'd like to keep in the loop about what's going on with SBA Brisbane, you can sign up to the mailing list up here or check us out on the Twitter feed. So today we've got today the SBA Brisbane talks are going to be COVID-19 themed, and we're fortunate enough to have two very accomplished speakers with careers in both academia and industry. And we're going to be starting off with Trent Munro from UQ. Professor Trent Munro is a senior group leader at the Australian Institute for Bioengineering and Nanotechnology and director of the National Biologics Facility at UQ. He's part of the UQ team developing a clinical stage vaccine candidate for COVID-19 as program director of the Vaccine Rapid Response Pipeline. His research is focused on the development, engineering, and production of complex therapeutics using biological systems. This includes engineering mammalian cells to improve their efficiency and utility in the production of complex proteins with a focus on biopharmaceuticals. Prior to this, Trent was an executive director of process development at Amgen USA, where he was involved in the translation of molecules from research through clinical trials and multiple commercial product launches. And he completed postdoctoral studies in cell biology and developmental genetics at Harvard Medical School at the University of Cambridge. So if you could all give me a hand in welcoming Trent Munro to the stage for his talk on COVID-19.
All right, thanks very much for the kind invitation, and glad to be here talking to you guys, and for the folks who are online, thanks for taking the time to dial in. So I'm going to take you through a little bit of a journey on some thoughts I have around what the intersection of sort of what I call traditional biotech potentially with SynBio means, and how at least I think about that, whether that's good, right, wrong, or indifferent, you guys can tell me. But before we start, I just wanted to highlight that last year, as many of you know, University of Queensland embarked on a program to try and look to see if they had a platform for a COVID-19 vaccine. And I'll talk a little bit around, the highs and lows of that program. But I also wanted to just highlight that at least at UQ, this was the group that did the vast majority of that work. And I wanted to make sure they got recognition for all the things they did, in particular, Keith Chappell, Dan Watterson, and Paul Young, who were really the inventors of the molecular clamp platform that I'll speak about. And then you can't see all of the things, but we also couldn't have done this without a really large collaborative group, both in terms of technical collaborations as well as funding collaborations. So I wanted to acknowledge all of those folks also. All right. So my understanding of SynBio is very rudimentary, I would say. So I feel like I've got a lot of educating. I need a lot of educating. I need you guys to tell me why this is so important and get excited about it. But I did just read this paper, and I thought it was really nice in talking around, there's technical aspects of SynBio, there's sort of education aspects of SynBio, And both of these work together in a unique way to let you create a cycle of invention that wasn't otherwise possible using normal molecular techniques. And I think that's really the way I see the power of this in general as a whole field. And you look at the way that it's changing communication or data or building circuits or building little machines, molecular machines, it's really driving a lot of innovation. So, things that you couldn't order by post a few years ago, you can. Things that you couldn't analyze before, now you can. So I think to me, that's the real power of SynBio as a platform. And I think we've got really clever at the way we think we can do molecular engineering, right? So we're crossing new boundaries, we're doing things all the time. But biology is really hard. And we all learn that lesson all the time. I'm going to tell you a lesson we learned about that today. So biology is really hard, it's really complicated. And just this week, for those that were following, I thought there was a really interesting story. So for everyone who's excited about gene therapy, I think we all are about the prospect of gene therapy. And there's some gene therapy programs on the market now. But one of the things that people were super excited about was our ability to modulate blood cells to look at some monogenic disorder correction. I think this is a really exciting area. I think one of the leading companies in this space has definitely been Bluebird. They have spent an enormous amount of money, energy on trying to bring forward an absolutely critical therapy. And this week was a really tough week for them because their clinical data indicated a potential imbalance in the presence of some issues with neoplastic cells. The really hard thing about this is that I don't think any of us will ever know whether this is treatment related, therapy related, or otherwise. But what it says is the complexity of trying to measure and do traditional clinical trials for these type of things when we're trying to program new circuits in people, it's really, really hard. So I think this is going to be a little bit of another bump in the road for gene therapy. And it also means for all of us, as we think around the technologies we bring forward and how we can advance this field, where should our mind be in creating better tools so that these kind of things become out of the question. If you get an imbalanced result in a clinical trial, you can provide a theoretical understanding for why that happens. So I just wanted to highlight that before I start. So I have this really boring way of thinking about how we use engineered biological systems to do cool stuff. And, people like to put labels and definitions on things, and I think that's fine. But the way I think about it is that we've got this real opportunity to use molecular engineering to drive a whole series of innovations across traditional industries. And that can be grouped under this holistic term of, say, a bioeconomy. Maybe the whole field is advanced biomanufacturing. And I really see that there are two major technology drivers for us all that we can use to progress these things. So one is synthetic and systems biology, which I think we've discussed. And the other is our ability to use cell factories and fermentation, or even acellular fermentation as a way of driving the production of those and looking at producing efficiency. And for me, the real impact of all of those is pretty obvious, right? Because you can span these different industries, whether it be new foods, whether it be new ways to power the way we get around, or whether it be chemicals that were impossible or really expensive or hard to get before, or whether it be things that I'm interested in, which is really the therapeutics of today, the therapeutics of tomorrow, and how we can use living systems to do really cool stuff to solve those problems for the hardest diseases that are out there. It's an overused and highly simplified example, but what let us drive the birth of biotech? Well, if we really think back, how do we go from using chemistry as our major tool to make products to now being able to produce very complicated molecules? And this is a large molecular weight compound, it's complicated, but we can even think of whole cells being the next step beyond that. But we just think what drove our ability to make progress in biotech, it was really the emergence of plasmids, the emergence of restriction enzymes, being able to do then heterologous gene expression, thinking about how we scale cells and grow cells, thinking about how we have technologies to even develop binding reagents, right? Like that was a huge part of this. And then, for some of these complex molecules, it was moving them out of, say, bacterial expression systems into other things like mammalian cell culture or yeast or bacular insect cell, whatever it is. But these things really then were the fundamentals of that first wave of biotech. But this is a long time ago that we started doing this. We're still reaping the rewards. We're still optimizing. We're still learning. But you have to cast your mind back. When people said that they could do this, everyone thought they were crazy. I mean, no one thought this was scalable. There was no economic model for it. And I think we're facing a lot of those challenges today for some of the things we're in. I think you just have to push through that, because really, this is one of the huge success stories. This is really where things work. And I've got two examples to show you why I think this became so powerful and why it did work. One is, if you look at the ability of complex therapeutics to erode the traditional pharma industry, it's kind of amazing. I mean, it went from a niche, very high cost sort of product, something that's now, accounting for nearly 1/3 of all products, or at least revenue on the market. So this whole idea of, changing the paradigm of the traditional industry, it's happening with biologics. And I think this is a really impactful slide to, as you think about, what we should be developing, what's the next generation therapeutics. What sits behind this is something much more complicated, and that's the payer landscape and how much are people willing to pay for drugs. And we're seeing that through some of the gene therapy things, cell therapy, et cetera, where the complexity of access to those therapies becomes really hard. And that's where I think we've all got a responsibility to think about new technologies to make those cheaper, faster, and better. But I'm not really going to get into that. But I will tell you, if you take an example of probably one of the classic monoclonal antibodies, it's a pretty terrible molecule, actually, for those that have worked on it. It was made sort of, got lucky, and then ended up getting even more lucky with some of the indications that went into. But this one antibody, AbbVie, has annual sales at the moment. They're on their peak, they're on the way back down, but we're closing in on $20 billion a year. Lifetime sales of $140 billion. I think all of us would be pretty happy if we developed a product with these kind of metrics. I mean, this is a unicorn biologic, but there are others that are doing the same thing. If people know about the checkpoint inhibitor pathways that are out there, the PD-1 agonist molecules, I mean, those are really doing the same sort of thing. So this happens in biotech. And this type of revenue drives excitement, and it drives investment, which is good. But it also means that, the bar for commercial success can be a little bit unrealistic. And I will tell you that people are trying to make copies of this, biosimilars of this product. And when you're up against a company that's turning over more than $1 billion a month in revenue, the number of lawyers that they can hire to find new IP is pretty impressive. So I don't know what the truth is, but word on the street in biotech is that AbbVie, they hired 100 lawyers whose only job it was to look at how they could develop new IP to defend from biosimilar competition. So I don't know if that's healthy, but that's an interesting story. All right, so I'm going to switch gears completely now and talk about the focus of this was talking about COVID. And COVID is the current thing we're living through, and we think it's pretty awful and pretty terrible. But the reality is, we live with infectious disease all the time. COVID's all the more terrible because it's impacting us. How dare an infectious disease come and attack our world, as opposed to people in many countries who don't have the same type of life expectancy and have to deal with infectious disease day in, day out. But anyway, that's the story. And this is an image, from I think 400 BC-ish of the plague of Athens. And for many of you who don't know this story, it's actually really interesting. There's a lot of conjecture. Obviously, it's a long time ago, and we don't actually know what happened. We don't even know exactly what the pathogen was. There's lots of ideas out there. But this was really a time when a lot of people had to pack in together, or were forced to, or did pack in together to try and deal with some conflict that was going on at the globe at the time. And it meant that concentration of people led to the emergence of certain disease agents, and really led to, the first recorded pandemic. So this is not new, COVID, and we've been dealing with it ever since. And last year, one of the papers that I read that I thought was one of the best papers, at least for me, to get context of what we were living through, was this one. And actually, one of the co-authors was Tony Fauci, came out of NIH, and it was published in Cell. And it was a really fantastic review of what led us to COVID-19. What was our holistic experience over many hundreds of years, or thousands of years on experience with dealing with large outbreaks of infectious disease. And I really thought this was just, it helped really solidify and sort of help me think about things and what we're going through, and sort of put it in context. So just put it up there to say, this is something we've been dealing with, and I'm going to talk about the tools that we have and what came out of this. But if anyone wants to go and look that up, please do. All right, so we got lots of tools out there that we use. We think we can make drugs. We're okay at that. Probably antibiotics is the best example we have. The rest of the things kind of work okay sometimes. Even like Humira is not that efficacious. But one of the most powerful tools we have, especially against infectious disease, happens to be vaccines. And it turns out they work really, really well. And there's many examples I could have chosen for this. I happened to choose measles as an example simply because it's an example of an unbelievable vaccine which gives you lifelong immunity against something that used to be just so commonplace in terms of causing severe disease. And so, it doesn't take much to say, well, that's a pretty impressive epi curve in terms of what happened to the disease. I will say, though, if you zoom in a little bit, what you can see, and this is some US data, this is the emergence of vaccine hesitancy. So I think, we all remember there was outbreaks up in the Pacific Northwest of measles in the US due to certain communities that where vaccination stopped happening. So this is just to say that it really does work, and when you stop doing it, it doesn't work anymore. Right. So when we think about pandemics, though, I showed you that big list and I showed you the plague that happens, but when we think about pandemics here for us, at least, for me, I kind of think of stuff like this, really awful pictures that are happening somewhere else. I don't have to really worry about it. But these things happen. I mean, Ebola is probably our closest example of a zoonosis which has persisted for a long, long time, with us just kind of hoping that it doesn't get here. I don't know if people remember the patient being evacuated into the US with a live Ebola infection, in the big bubble and all of that drama to get that patient there and look at treating. But this was happening, and one of the biggest outbreaks, 2014 to 2016, the world was just kind of sitting there watching, oh, look, it's another Ebola outbreak, lots and lots of people are dying. But luckily for us, it did stir some things into action. And I think one of the best things to come out of that was at least a harmonized approach of putting some additional money into funding vaccine research, especially for emerging neglected pandemic diseases with pandemic potential. And that organization was called CEPI. I think many of you have now probably heard of CEPI or read about CEPI. Probably not so many of you knew or heard about CEPI before COVID. But they got a couple $2 billion together to really look at how they should invest. They picked a few different areas, so MERS, Lassa, Nipah, chikungunya, Rift Valley fever, as areas of sort of interest. But I think one of the coolest things they did is they said, everyone's been talking around this disease X idea, the thing we don't know. Maybe we should really fund some platforms where we could look at rapid response vaccines that could help us if we ever had a disease X type situation. And so that's what they did. What happened out of that was that, of course, as we know, this was a slide that at the beginning of last year was kind of, we're still processing it. I think we're well and truly sick of it now. But, there was the real case disease X that I think all of us were fearing actually happened with first reports back in December 2019. And of course, we all know the story from there. This was SARS-CoV-2 and the resultant pandemic we're all living through. And so I think the interesting thing about that is that all of a sudden turned our attention from the idea of something where, it was far away, didn't really relate to us. It looked like it was happening in a village somewhere that, many of us don't know, to something where, this is a disease that can easily overwhelm healthcare systems. So if you look, you've got one patient here, and you've got, whatever it is, 6 or so people desperately trying to help this person from the disease and not expire. And you can understand doing that number and that metric, and you look at the amount of gear and you look at the amount of stuff that goes on, how pretty easily, even with low mortality rates, if we consider SARS-CoV-2 to have a low mortality rate, it can quickly overwhelm our modern healthcare systems. And so then what was the obvious thing? And so it's what everyone was asking back then. It was kind of like, well, can we make a vaccine against this? We sort of worked well with other things, what can we do? And I think it was really interesting for all of us to think around, what that meant. Could we do it? There's all that talk, well, there's never been a vaccine for coronavirus. I'm not sure it's possible. And, all the experts out there saying, how long would it take? And I know whenever we were asked, we always gave a figure that, was never right or was always too fast or too slow. So that was a question that was on everyone's lips at that point. And really the only thing we had was historical data saying, it could take a really long time. We just don't know. I mean, some vaccines, some diseases, of course, many diseases we don't have effective vaccines for. And the ones that we do, the vast majority of them, it took a really, really long time for us to go from, starting development of a vaccine to having an effective vaccine. So that was kind of worrying. I think what we'll talk about is the last one down here, which is, of course, how quickly we've done it for COVID-19, which is great. But, that wasn't a data point that we had at that point last year. And so we know that we've got a bunch of different tools that we can use as vaccines, and these all have pluses, minuses, complexity, etc. But these were really, really well defined. And so people, as soon as this happened, went out and started deploying all kinds of things across these different broad areas to say, okay, what are the tools we can have in our toolbox to think about potential COVID-19 vaccines? The way I think about this, actually, and it's just highly oversimplified, is just to say, well, we kind of think about it. We've got the really sort of old school vaccines. We've got the new vaccines, and all of them kind of require some form of biotech type knowledge. But you can really think about it as like the very traditional biotech, like the idea of just growing up virus, analyzing it, and just using that product through to, hey, we can actually reprogram cells using, modified nucleic acid to get them to make what we want, and maybe that's the solution. So you've got this continuum here, and I think just wanted to make that point that, everything was on the table as we went forward. And of course, there's a lot of stuff behind this that fed into the way we make these that also is applicable to those broad categories. We as scientists always think that we're pretty clever and we can predict the future and we know what's going on, and we always think that we can sort of talk about, where we're headed. What I think is that there's very few of us who've got any prediction right in the last year. I mean, mostly we've been abundantly wrong about pretty much everything, with the exception of maybe a few epidemiologists who just said that this virus is going to go across the whole world and a lot of people are going to die. A lot of the other scientists were wrong. And so I like this quote that I listened to last week from Paul Offit, who, if you don't know who Paul Offit is, he's one of the most well-respected vaccine experts in the US. He helped develop a rotavirus vaccine. And what he said was, if you asked 100 scientists back in January 2020, when we first published a sequence of SARS-CoV-2, do you think it's possible in 11 months that we're going to have, 2 clinical trials completed, one of 30,000, one of 44,000, using a novel technology, i.e. mRNA, which we had no commercial experience. And we're going to find that those trials for this disease essentially showed 95% efficacy across all age groups, all comorbidities, with a vaccine that apparently is safe. I don't think anyone would have gone out there and swung that bat. Luckily, some people did. And we're very fortunate that that's one of the tools that we'll hopefully have. There's many others, but it shows you the danger of prediction. It shows me the danger of prediction and trying to decide, what platforms are good, what technology is good. And really, the difference is that all of a sudden this technology went from something that people had been shouting around for a long time to something that just happened to find the right use, and we threw billions and billions and billions of dollars at it, and we got to advance it really fast. And I think full credit to Pfizer and Moderna here, both on the ability to produce that molecule and also do clinical studies, something very, very special. So the other really terrible thing that's happened is that data is so available to us. So, I got to the point where I just stopped loading this web page, and maybe some of you did too, because it's just kind of got too depressing. But it is kind of amazing the types of dashboards that have come up, and I just wanted to say that whether it's looking at our ability to track waves, country-specific data, whatever it is, the availability and the immediacy of data has kind of been amazing. So, that's one part of scientific progress that we've had. I think there's a number of other really amazing things that we think, as a scientific community, what have we achieved over the past sort of 12 months or so? We went from having a novel virus emerge, its genome being available back in January, to the point where there was over 2,000 publications a week on SARS-CoV-2 coming out, which kind of blows my mind. I think it was over 90,000 I last looked. It might be 100,000 now, I'm not sure, but that is an incredible amount of literature to sort through and figure out what's real. Hundreds of vaccines in development, hundreds of potential therapeutics in development, of which now there are multiple vaccines actually approved out there. I think when I looked at this the other day, there were more than 200 million vaccinations already given across the different platforms. Obviously, we need that to speed up, but that's pretty amazing. And we're tracking the genome of this thing in real time as it moves to essentially a naive population. The problem with that is everyone's a little bit obsessed by variants at the moment. The impact, I don't think we know. I think, we'll see. But this is our first real ability to look at a genomic level. What happens as a virus spreads through a population? So, if there's anything evolutionary biologists are going to learn from this, it's going to be a treasure trove. So from a scientific perspective, unbelievable progress, and it really shows what happens when the scientific community really throws its focus towards a problem. And I should say, for those who've been reading some of the literature, some of the literature and progress in the science and this on the immunology side, structural biology side, to me has just been mind-blowing. There were things happening every day that I didn't even know was possible. Of course, last week for us here in Australia, I think we're all kind of happy to see this plane land, and for these first shipment of vaccines to come off. And of course, Monday we'll see how that goes, and that'll be the day that we start our vaccination program here in Australia. But who would have guessed, if you'd asked again on predictions a couple of years ago, Sydney Airport would be tweeting around a Singapore Airlines jumbo coming, or Singapore Airlines, I don't know what kind of 380 or whatever, bringing in plane loads of frozen vaccine, that would be such an important milestone for the country. Okay, so I guess the question for us, and I'm going to focus a little bit on some of the work we did last year. Did SynBio focus tools impact our ability to develop a vaccine at pandemic speed? And I think the answer to that is definitely. The answer is, could we do more, and are there really amazing opportunities to do more? I think yes is the answer to that as well. Really, luckily, as I was thinking about this talk last week, this paper came out, and I thought, I could just kind of talk about this paper for a whole talk, but that really wouldn't be fair. But I will say it was just kind of interesting, as we're all thinking around what these tools mean, other people are starting to think about this as well. So just another Cell paper that came out talking around, what their view, I think this is a Harvard MIT paper, what their view was on, what SynBio has done in the COVID era, for thinking about how it's enabled a future technology. So I'm not going to speak on that, but I just want to say it's out there in case people wanted to pull that up and they didn't see it. All right, so I am going to talk a little bit about the technology we were working with, and it's all about molecular machines and how we dealt with designing sort of complex things. So the idea is basically that when a virus interacts with its receptor, it's in this unfolded or post-fusion conformation. And that really, that's not the form that we want to raise antibodies against. We really want to raise antibodies against the compact form. The idea is that there's a better chance of getting neutralizing antibodies against this form. But when you make the protein in isolation, it tends to flip into that other form. So the idea is, if you can constrain that through the use of some kind of domain, in this case a trimerization domain, that maybe that forms a better antigen for vaccine design. And that's the whole concept of this molecular clamp. As you'll all know, the particular clamp we use is a 6 helical bundle that's derived from GP41 and HOV, but it works really well. So this domain is able to basically lock this viral trimer into the form we want. We can look at it on EM, we can show that it's in the form that we want, and we can use it as a purification handle. And that's really important as we were thinking about how do we produce tens of millions, hundreds of millions, or even billions of doses of this particular product. The team had been working for more than 10 years on this technology and looking at its ability to work across a whole range of different viral antigens. And they submitted a proposal to CEPI a couple of years ago saying, hey, we think this could be a good platform for rapid response. And they got the funding from that, and that was really the start of this journey. Of course, it's not the only technology out there. There are other ways to look at stabilizing these molecules, many different approaches have been tried. One of the other really elegant approaches involved knowing a little bit about the structure and engineering in new amino acids, the so-called 2P mutations, where they're able to engineer in additional prolines to do the same type of thing, to lock the molecule into the pre-fusion conformation. This team had done this with MERS a number of years ago out of NIH in Texas. And of course, the good story for that is that actual construct now is the foundation for a whole bunch of these vaccines that are either approved or on the cusp of being approved. I think the one notable exception here is AstraZeneca doesn't use this particular technology for theirs, and that's because they're able to show that once it's expressed in the cell, they keep the transmembrane domain. The vast majority of their molecule they believe remains in the right conformation to elicit an immune response. So we had this program, we were on our way, we were thinking about, taking this into the clinic. We were going to do some studies on influenza and MERS and RSV, and then we were going to do disease X a couple of years from now. Long story short, last year, of course, with COVID, we threw all that out and we said, could this be applicable to SARS-CoV-2? And I'm not going to trace all the bits of that journey, but just to say that essentially what we did was the UQ team worked with a whole bunch of partners, but the most important of those is really forming a commercial partnership with CSL. And CSL was an amazing partner, just as they are right now manufacturing the AZ vaccine and for release in a couple of weeks. They pumped out millions and millions of doses of this vaccine in an accelerated fashion. And it was a very fruitful partnership and really built on the experience they have in recombinant mammalian cell-based proteins down there in Melbourne. So that worked really well, really fast progress. And it also relied on a network of capabilities we have here in Australia. We're able to do some biologics development, ourselves up here in Queensland, of course, the SynBio team down in Parkville and Clayton, really enabling to this whole effort. What did we do? So we had to screen a whole lot of different molecules. And I think, again, when you're trying to do computational screening and add different parts of a molecule together, I think this is a sitter for a SynBio type of approach. We did this sort of with brute force. I think this is an obvious area where using molecule design, molecule assembly, rapid screening would be really amenable to a SynBio type of approach. And we did do some work on that. But this molecule is pretty complicated, so there's a little bit to do there and learnings from that. The other thing that we had to do was really think about how do we do large-scale manufacturing and how do we select individual cells which are going to be suitable for that manufacturing. Again, I don't have time to go into all of this, but we're able to form some really important partnerships with a bunch of different biotech companies out there to let us do this. And so the ability to really, from us going from making, 10 mils in a shake flask in our lab through to 2,000 litre bioreactors at CSL relied on really fundamental platforms and technology that were from some of these companies. So we're very grateful to them for letting us make that leap. Of course, one technology which I've been working on for a number of years and I just think is pretty cool is this ability to use essentially nanofluidics and combination with driving cells around with photons to select for individual cells really quickly, get those cells out because they're expressing a gene of interest, and then go on to do some study. And Marianne, who's here in the audience tonight, really was the driver of this machine and under immense pressure was able to use this. It's really our first program at UQ to use this technology. Really did an amazing job on the phone to San Francisco every day with the company working out how we were going to do this. I think they've even taken one of your pictures now, Marianne, put it on their website, and we developed a custom assay at the time. So, just another really great collaborative effort. And this was a technology that wasn't available a few years ago, but is now that's something that's really able to do something that usually takes quite a long time and actually do it much, much faster. So the point of all that, of course, was to say, what does manufacturing really look like? And so in our lab and the SynBio lab, we're able to use that 50 litre sort of keg-sized device there on the left. But what we really needed to be able to do was move to the 2,000 litre kind of device over there on the right down at CSL. And we were able to do some calculations of how many doses we think we get out per run. And we thought we were probably up to the 10 million doses per run, which I guess is okay. We probably would have been really happy if we were up to sort of 100 million doses per run. But again, this was something that became, it's something that happened. And I think all of us were nervous of whether this was going to be doable or not. But it shows the power of using these tools that we have, moving quickly and then going into the scale-up phase. So I'm just going to finish with saying, well, what happened? Why aren't you lining up for the UQ vaccine on Monday instead of the AstraZeneca vaccine that probably most of us will be getting? And that's because our program was halted back in December. We now join a small group of vaccines that have decided not to progress, most notably those from Imperial College and 2 programs from Merck. The Imperial College decision was a commercial decision. The Merck decision was a biology decision. They didn't believe they had enough strong phase 1 biology to support the progression. Ours is because the clamp domain, which I showed you, actually the immune response against the whole molecule was so strong that we also developed an immune response against that clamp domain. And because it was derived from the GP41 molecule, it turns out that GP41 antigen is also used in a bunch of third and fourth generation point of care and lab diagnostics for HIV. And so what it means is in some of the screening assays that you would have, if you had the vaccine, you would essentially come up as a false positive. So we've been working with HIV experts in the community to look at if that issue was actually navigatable. And just to show you the signal differential, if you look here on the left on this graph, that's our vaccine response, and this is a HIV test. In the middle is a bunch of examples from Queensland Technology on what a HIV positive individual looks like, and on the right, what a HIV negative individual looks like. And you can see while the signal is low, to blow that axis right up, there's a bunch of overlapping samples there. And so it definitively means that either on a stick-based test or on somebody's lab immuno test, you would get this false positive result. And so in the end, the decision was, as we saw other vaccines coming forward with fantastic efficacy, if our vaccine was in any way responsible for vaccine hesitancy, I think that was a too big a risk. And so the worst situation was that, this became something that stopped someone somewhere getting a vaccine. I think we all felt it's in the back pocket. Anything happens to the other platforms, it sits there, but really that's not our focus. That decision was made, and it was a decision made by the Australian government in consultation with CSL. But I will say that our clinical paper is coming out in Lancet Infectious Diseases. I think it's online now. And we were really blown away by the clinical results, and as were most people who've looked at it. So from a biology point of view, clamp platforms validated, frustratingly well validated. And this is just an example of what the immune response looks like for some of those phase 1 trial individuals. This is for the 18 to 55-year-old group. We also have the older adult group. That data wasn't unblinded at the time. It looks really good too. The safety profile looked as you'd expect for an adjuvanted protein-based vaccine. So very, very good. And really, the fact is that people produce the type of immune response you'd expect to provide protection. We have no way of saying whether it would provide protection because this trial is obviously not powered in any way to look at efficacy, and that was really the next step for us to go into a 30,000 participant phase 3 study that won't be going ahead. So that was the story for us. What are we up to? Well, I think it's an example for us of the cycle of what happens when you go through an engineering cycle. And right now we're continuing to be supported by CEPI. And a few of our other founders. So we're going to use a bunch of the money from last year to continue on and look at CLAMP 2.0 and CLAMP 2.0 in the context of having a protein-based adjuvanted vaccine for COVID, as well as looking at what the situation is with some of these variants as COVID settles in to becoming an endemic disease in the population. What are those variants? What does the next round of vaccines look like? As well as, what does the booster regimen look like? Are we going to need this vaccine 1 year, 2 year, 5 years at all in the future? And those questions remain unanswered, but definitely having an adjuvanted protein solution for it is one way to think about that. So we're going to be busy pursuing that this year, along with some other vaccine work as well. So with that, we've gone well out of time, and thanks for listening, and hopefully that was helpful. All right, thanks, bye.
Yeah, stated in your talk that you said it rather
So I talked about the danger of prediction, but I think how long it'll be around for, I don't know, but it's going to be around for the foreseeable future, just as it goes through the population. I don't think it's going to go away. Whether natural immunity in places that don't have access to vaccines cause that to be a diminished presence of the vaccine, or if vaccines are able to greatly diminish the presence of the virus so that it's no longer a major cause of hospitalizations or death. We hope that's true, but it is possible that some elements of this could become, a recurrent vaccine, recurrent disease, probably in a seasonal type fashion. But again, we don't know what we don't know. So.
Thanks for talk, Trent. I was just curious about your perspective on how does a business model work for these big pharma companies making this medicine? Obviously, they've got a lot of public investment, and obviously governments can be buying it. How do they determine the price?
Well, there's no money in vaccines, is what everyone will tell you, and that's why we don't have a lot of good vaccines. But just yesterday, I think the estimates came out for what their revenue expectations are for both Pfizer and Moderna. I haven't seen the Astra projection. So I think Pfizer's predicting $19 billion of revenue in the next 12 months, and Moderna $19 billion. So we'll see, how long that goes for. But I guarantee that's an ROI. So we'll see what that looks like. But yeah, it's really complicated. I will say that I really honestly believe that AstraZeneca, Pfizer, and Moderna, Novavax, and J&J did this without running those commercial numbers. And that's good. Merck did run numbers and decided to stop their program. So we'll see.
Following this question that Henry made, and showing how fast was the response to this vaccine, in which way that response is associated to the public interest that was a huge economic burden for that to happen, right? And we got in 1 year of this vaccine. So based on those results, do you see soon all that learning being implemented on vaccines in other diseases like HIV? Or it was happening because the nature of the virus itself?
Yeah, I think we got really lucky. I think if this was influenza or HIV type virus, we wouldn't be in the same situation, even if we threw lots of money at it. It's the same reason why we don't have great cancer drugs for solid tumors, for example. Those problems, we're probably just not at a state of maturity from a biological perspective to solve them. But it just so happened we took the most obvious molecule from this virus to stimulate an immune response. And so far, we'll see what happens, but so far, that looks efficacious and safe. So there's both elements to it. But the mRNA platform, that's made huge strides. I think our understanding of disease-transferring immunology of these types of viruses has exponentially improved in the past 12 months. And I think those learnings will flow through. How long that takes, I don't know.
Thanks for talking about that. I was wondering, do you think it's that for the last few chapters what I looked at vaccine response in countries? Do you think only get this level of response by vaccinations?
Yeah, I mean, we can't eradicate polio, so, because of wars in Pakistan and Afghanistan. So I would love to say that it would. I don't think it will until the problem becomes a Western world problem. We're just not interested and we won't do it. So that's kind of a sad state of affairs. My view, though, on the response side is that I really think we should be thinking about how we deal with infectious disease more like how we deal with defense. I mean, why would you not just dedicate proportions of rich country budgets to, medical countermeasures or, pandemic response? It just seems to be a no-brainer. I think that we have really short memories, and in 3 years from now, we'll probably be back to the same baseline we were in, the same vaccine development scientists be going, hey, we could, develop a vaccine against this if you just give us our grant. I hope not. Again, I'm predicting, so we'll see. So are you, Kirill?
Do you know how to get rid of the Zoom bar? It's on the screen.
Yeah, if you can hide the bar.
Yeah, that'd be fantastic.
If you can, it's that was great, Trent.
Oh, yeah, no problem.
Oh, yeah, no, go ahead. Yeah, I'll like to hear myself.
Okay, yeah, sure. Thanks again, Trent, for a great chat. We really appreciate it. Now I'd like to introduce Professor Kirill Alexandrov from QUT. Professor Kirill is a CSIRO QUT inaugural professor of synthetic biology at the Queensland University of Technology and a CI of the ARC Center of Excellence in Synthetic Biology. His group is interested in artificial sensing and signal transduction through proteins, two-way connectivity between biology and diagnostics, and the development of approaches that allow rapid and flexible production, engineering, and analysis of proteins and protein complexes. Kirill co-founded the German biotechnology company Jena Bioscience in 1998 and the UK-Australian synthetic biology company Molecular Warehouse in 2015. Before joining QUT, he was at UQ in the Institute for Molecular Bioscience and the Australian Institute for Bioengineering and Biotechnology as an Australian Research Council Future Fellow. And he was a group leader in the Department of Physical Biochemistry at the Max Planck Institute in Dortmund, Germany. So, yeah, I'll take it away. Just maybe, can you give me a second to try to figure out this bar?
I can start talking already.
Great.
It's just not to keep people away from their drinks and food and socializing. Yeah, so thank you so much for the invite here. That's actually quite a great coincidence because we have done a fair bit of work on COVID, and I think it's actually great that it's a back-to-back feature with Trent because first of all, Trent world is the vaccine world, we're doing something different. But also it's a very different corners of scientific enterprise or academic enterprise because the just amount of funding that went in those efforts, so what we've done was done on the ground of $49,000 from Stanford University. And Trent, what is it, $30 million? $20 million. So scale it down, set your expectations accordingly to what you're going to hear. Yep. Yes, victorious. All right. So I mentioned vaccines briefly, but again, it's let's see if I can actually. Oh, doesn't. Okay, I will have to do the manually. Right. So no, I can do it manually. I think it's one way or another. Good. Oh no, it's actually frozen now. Sorry, Zach. I think we just have to live with it. I'm sure there is a. If we can we try hiding the video panel again? Yeah. Okay. Oh, yes. Both the best of both worlds. All right. So the world of pandemic, I think it's just amazing how quickly it all happened. It's like almost like the life of Facebook and Snapchat. It just all happens in a matter of months. It's all in fast forward. But it took everybody by surprise. Everybody was shocked. The world was collapsing. Stock markets collapsing. Civilizations, borders closing. The world was going into a state we were not familiar with. And, not surprisingly, there were people over time that have expected, predicted, SETI was one of them. But majority of us, Trent said, were quite complacent, despite some major promotions, that were great box office hits, and very powerful world leaders flagging this problem not so long ago and actually investing their hard-earned dollars in solving it. And now after I flagged those, actually I'm very proud to show my own slide from 2017, where I was summoned by Non-Proliferation Conference to Buenos Aires to talk about the possible threats and what synthetic biology would contribute to the dark side.
And the only thing I could really come up with would be better viruses. And just because of and you can see it on that slide. This is the same discussion. You don't do anything, you have high deaths. You do something, you have huge economic impact. And then so I said that, everybody listened up, and then the train moved on. We started talking about hand grenades and talking about gasoline, how much more dangerous it is in a pandemic. Well, one thing that we clearly learned from our pandemic experience, that public health measures work. If you do things by the book, they work. Lockdowns and quarantine work at a staggering economic cost. And the number that I really shocked me. So this is, for instance, the U.S. government debt as a percentage of the GDP. So the U.S. has now reached the government debt that of the Second World War, so the existential crisis of the entire civilization. And in the pandemic, 9 months of the pandemic, the U.S. government pumped more money into the economy than in 5 previous recessions. So this is absolutely unprecedented, and we haven't really started to understand the impact that it will have on the economic system. So, and in other metrics, the 5 days Melbourne lockdown, which everybody welcomed, costs more than this tallest building in Melbourne. So, for every day you get like 1/5 of it down until it disappears. So, what if you think about the playbook of pandemic response? What is the playbook? And it's actually very simple. First and the most important rule, actually know that there is a book. And it was shocking how many countries didn't know how to do it, or didn't want to do it, or didn't know how to manage it. And the first thing is, of course, preparedness. And that's where we scored very badly. And preparedness is very simple. First of all, you have to know your enemy, because they don't come from nowhere. The viruses are circulating in the ecosystems for millions of years and mutating, adjusting, changing hosts. Surveillance is extremely important. I talk about it a little bit more later. So you have to know what is out there, what can jump, and what had jumped, because many of those jumps actually happened before, and then the virus comes away, died out, resolved, mutated. You also need to understand how the viruses do it, the mechanism. That's why we were so spectacularly successful with COVID, because it was related to viruses that we knew, understood, that we worked with, tried to make vaccines against, tried to make drugs against. And so it was very clear what to do. And that is, of course, shortened the timeline very significantly. And the chapter 2 of that very simple book is the response. And what worked in the countries that were successful is really 2 things: testing and public health measures, such as isolation. And that's all there is to it. This is old as the world of infectious diseases, and you just have to follow the rules. There are, of course, new elements, and I'll talk about them a little bit later. But for those who are interested, I highly recommend this podcast. You probably cannot see it on the screen. It's a podcast run by Lex Friedman, and just type in the Google Lex Friedman diagnostics, and will come up an amazing podcast about the coronavirus testing and testing in general, and the impact it can have on public health. And I'll come back to it in a second. And this is step 1 of the response is develop vaccines and drugs. So we're worried about the vaccines. So now vaccines are drugs, because it's probably both. It's not either or, but you don't know. There's a fog of war. You don't know what will be faster, what will be better, what will be possible. And just to remind you the context, this is August, and the WHO says, oh, we're actually not sure whether there will be a vaccine. Yes, there are vaccine trials, but we don't know. Maybe, as Trent said, maybe there will never be one, maybe they will be inefficient, maybe they won't last. And that could have happened. And so then you have another alternative. You can go and start making drugs. This is, of course, a much less attractive proposition because drugs take so long because you're starting, in most cases, from the first principles. But that actually has been done, the groundwork for it was laid fairly early. So that's the study from March 2020, where people done proteomics of protein-protein interaction of the COVID virus. And so they just took the open reading frames of the virus, transfected them in mammalian cells, did pull downs, did mass spec, did proper filtering, and said, well, how many proteins this 29 open reading frame viruses interact with? And they said, oh, there are almost 400 different proteins. It is a very complex virus. It does a lot of different things. Most of these interactions we don't understand. But they said, oh, well, at least, some 65 of them may be directly or indirectly targeted because there are some approved drugs. Then who knows, if we use this drug or use the derivatives or change the regime, maybe it will help. So that was the gist of this paper. The problem was, it's not even a problem yet, that was a great first step. So the next step there is actually, so just go back. This is inferred data. So this is all inferred from mass spec, none of these interactions are validated. There are some biology that in some cases corroborates those findings, but this is far away from actually starting a drug development program. So that's, and of course, just even validating that number of protein-protein interaction, understanding the affinities, kinetics, understanding what structures they are, what you can potentially target, is a gigantic task. And that's why we got attracted to it, because we really like developing technologies for solving very difficult problems, technical problems. And we teamed up with a couple of people from the US, and that was Dmitry Petrov and David Enard who are computational biologists. And so what these people did, in the early, after this paper came out, they asked a question: has, we know now the genes that viruses use in getting into the cell. Has there been precedent in human evolution that all of those genes started mutating faster than average? Because of course, all of our genes, mutate, accumulate mutations all the time. There is certain mutation drift. But if there is evolutionary pressure, for instance, if you are a viral entry gene, and if you get a virus, your survival chances decrease. If you have a compensatory mutation or advantageous mutation, you're likely to survive. So that's the question. So if we take this network that was computation identified and look for what's called gene sweeps, the increase in the rate of mutations in all those genes, we look in the human genome, maybe we'll see whether the virus came around before. And so we've done the analysis, and what came out is that the virus actually has come through the human population before, particularly through the Asian population. So there were two clearly detectable pandemics. So one is about 400 generations ago, and another one about 900 generations ago that left very significant genomic traces in the genome. So, and they identified about another subset of about 60 proteins that mutate particularly actively in those events. And so we were very interested in these two subsets, one of the genes that could be targeted and that the virus is using, but also the human proteins that mutate fast, because if you mutate fast, they're obviously important for the host viral interaction. That is another avenue how to identify the targets that may be drug. So So now we have to convert that from computational data into the robust biochemical assays so we can lay the ground. And giving you the context, that's where I'm running around, literally and figuratively around the world, trying to get any money to do this work. And so the only, and it was like zero, it's like dead, and they went less successful fundraising campaign than back then. So the old, we finally vitro organized this like an internal Stanford grant rise, it was $49,000. So we actually could do this work. And, to be able to do that, we actually have to get all these products made in the form and look at them in a test tube, so to understand how they interact. And there are two main ways of making recombinant proteins. One is in vivo production, that's what vaccines producers are using, most of us in the lab. And then there's an alternative route is in vitro. So in vivo production, you take an organism, you stick a piece of genetic information, you reprogram it, and it gives you the recombinant protein. In vitro, you do similar thing, but you take the organism and you mush it up and you throw away everything which is not related to the protein synthesis. So you basically convert it into a protein polymerase. And then you reprogram this polymerase with your DNA or RNA, and you get your recombinant protein. Why is it a good technology? Well, it is attractive for a couple of reasons. One, there are a lot of limitations what you can do to a living organism. The living organism can die. And the more you modify it, the less happy they are, the more likely that they will keep the buckets, start going slowly, start modifying the gene. Obviously that doesn't happen with in vitro translation reaction because it's already dead, so you cannot kill it, so it's always happy, and so there's no limit on number of genes you can put in there, type of genes, or type of chemistry you put in the reaction. The second advantage is, of course, the timeline, because you're not dealing with living organisms, you're just dealing with chemical reactions, which are quite fast. And so you go from days that you need to make a recombinant protein in a living host to hours to make it in a cell-free system. It comes at a price, and the price is the yield. The yields are much lower. You get homeopathic amounts of proteins. But you still can do quite a lot with it. And it's an old technology. It's been around for since the discovery of genetic code, and people developed many protein expression system, cell-free protein expression system based on different organisms. And I'm not going to go through them, but they're basically there's systems based in prokaryotes, and they are very productive, but make often trapped proteins, and they very systems that are difficult to make and not very productive. And we contributed to that, say about a decade ago, a system based on the work called Leishmania tarantula, which is a parasite of lizards, which was isolated in culture about 120 years ago, and lives very happily ever since. And so that is, we like it because you can grow it on a large scale, and there are some details of molecular biology that make it very suitable for what we're trying to do. We created a vector system, which is called PM cell-free. So it's a gateway vector, which will drive transcription and translation in any cell-free system. So it's completely host independent. You can use it in E. coli, you can use it in mammalian, you can use it in plant cell-free system. And you also can transfect it into the mammalian system, and it will just work as a marker vector. So, and it's compatible with the gateway. And we like gateway because it allows us to connect to human warfare. The large collection of human vocal reading frames in the gateway vector systems. So just very briefly, we've done a significant amount of benchmarking on systems. We know we're using different methodology, and this is just a gel showing you that if you take a set of genes, you express in different systems, you have different outcomes. And you can see that in E. coli, large genes just get broken down into pieces. I'm actually not going to show much of this data. Basically, Trust me, because I want to make other points today. Yeah. So I'll just go down. Yeah. So basically, important links that we benchmarked very extensively. The Lithuanian self-risk system is as good as mammalian self-risk system. We develop a system for protein-protein interaction analysis based on the self-risk system, because we can take these vectors, we can clone our genes with fluorescent tags, so they express as in either green or blue, green or red, and then we can capture them on the beads of alpha screen systems. So there are two types of beads, the donor and acceptor. And if proteins interact, they bring these beads in proximity, and you get a signal, and you can use that in high-density form and tradable format to perform the assays. And you get high-density data. So we actually originally started working on the viruses with Zika when Zika came around. So we piloted that with Zika. So Zika is a small virus. So we made all 9 open reading frames. We translated them in cell-free, made them into 2 colors. So that's actually the whole proteome of Zika there on 1 gel. And then we've done the complete cross-match, looking at the interaction within the Zika virus, just to see what subcomplexes Zika virus can assemble, because those are often biologically important. And so that's the map that we were able to generate in about space of about 2 weeks when we got to it. That is the pre-existing map from the literature. So we discovered 30 new interactions within Zika virus. So when it came to COVID, I'm not going to go through a lot of details, but so this is again, the whole proteome of COVID. Those are synthetic genes cloned in 2 different vectors. Again, red and green. Then we were able to make, by now about 120 human proteins. Again, in 2 colors is only a subset. And we went and those are co-expression. We expressed viral and human proteins together, and then you do the proteomic analysis. And then you can go and start actually validating those interaction maps. And so if you end up having particular nodes, and then you get the different series of signals, and you have a certain threshold to know what is specific and not specific. And I'm not going to go through the individual interaction and just show you that that's where we got so far. So we validated a lot of its interactions. So in this assay, the alpha screen assay, is actually industry screening assay for chemical compounds. So you can take this assay and you can plug it directly in a robotic compound with the robotic platform with the chemical library and start screening. And we do have collaborations in biotech where people use cell-free as a delivery pipeline to drug discovery. So just a couple of words, about functionality, because interaction indirectly tells you something about functionality, but there are some couple of bits that quite well characterized, and one of them is the interaction of the RBD receptor binding domain with ACE2 receptor, which is the gateway for the virus to get in. Something that we tested, in our system. So this is again an expression of chair ACE2 and GFP RBD, and they interact beautifully in the assay. And it not only interact in the assay, you actually can see the action of the antibodies in this assay. So if you take this assay and you drop there the antibody, like this is anti-RBD antibody, which is bought at 5 nanomolar, they completely knock down the signal. So here we clearly can make a complex functional proteins. Now, very simple conclusions before I start speculating again. So I think we showed that LTE system has a potential for doing large-scale analysis of mammalian viral proteins. We have an interaction platform, and you can use this platform for analysis of effects of small molecules antibody on the interactants. I think there are some important lessons that we learned, that I think quite important for viral preparedness. So, we believe the cell-free platforms has a lot of potential, but they are actually not the technical bottleneck. The technical bottleneck turned out to be the large-scale DNA synthesis and cloning. So we spend 3 times longer on getting our hundreds of constructs sorted, synthesized, cloned, prepped than we anticipated. We since developed a different pipeline for DNA prepping or amplification for cell-free. But, particularly in the world which is disrupted by a pandemic, this is actually becoming very, very difficult. And that's something, if you think about the preparedness, that's something you need to have under control. So you actually, because if you go to a website of Twist or G9 or Genie University, say, oh, no problem, mate, 7 working days, 9 working days, pay more, you get in 3. Forget it, you're going to get the last Genie you're going to get in 2 months. Because there's, it is a technology which is only 90% efficient. And that basically screws your ability to do this large sets. So that is important. It turned out that repositories of human genes that we use, and there are 2 of them. There's one in Arizona and the second at Harvard. They're a mess because they're run by academics on, I don't know what type of funding, but first of all, they shut down for pandemic. And then it turned out that they actually didn't know where the clones were, and then they sent us the wrong stuff. So, given how essential this resource is, being able to access it is actually very, very important. I don't think globally we have one. So the only option you have left is going through DNA synthesis and do everything to Noah, where again you're running into the same sort of problem. So if you think about pandemic preparedness, this is something to have a very, very good look at. So, and it's like with a car, you can have a super BMW, but you have a flat tire, it's not going anywhere. And so that's why it's important to pressure test those processes and have a worst scenario simulation. So what happens if there's no FedEx? Can easily happen. That's it. Are we done? Is civilization going to die? Or you actually go drive somewhere and breathe. And this is particularly important in the Australian context, where we're so dependent on overseas technologies. Right. So last bit, which is loosely connected to what I was saying, but it's connected to the self-free. So we were talking in the beginning, I was saying how to deal with pandemic and how important the public health is. One underutilized resource that most countries haven't utilized, and Australia haven't utilized, and US is starting utilizing now, is really self-testing, for infection. And testing particularly for viral load, because there's gold standard, there's a PCR that is done in a central lab, which is very accurate, takes a while. That's why, in Queensland, where we have very good services, you get the answer in 24 hours, maybe you get it in 12. That is important, but more important is to know, are you going to infect other people? Yes, it is important to know if you're sick, but the most important answer for the public health is actually how infectious this person may be. Therefore, and that is determined by how much virus you shed. And there is actually a fairly narrow window in your disease when you're super infectious, and everything left and right is a lot less problematic. So if you even have not a very high sensitivity, but you're doing it repeatedly again, your outcomes will be very good. So you actually may not have to shut down the economy to quarantine 99.9% of healthy individuals, as we've done just now in Melbourne. You can just do sweeps and see, if you start thinking how often you pick up signals and the signals are increasing. So to do that, those are fairly simple technology. Those just testing for viral protein. And again, now it's a bit better resolution. So again, go and listen to the talk, because they talk for 2 hours about the technologies, about the impact, about the interaction of public health and point of care testing. But it has to be cheap enough so it can be offered for free every day, and that you get like a pack in the mail every week from the government for the duration of the pandemic, and just to every bar there is a balancer that just swipes everywhere. Then it will work. Then you will control the pandemic, and you can continue operating largely as normal. And so that's where the self-resistant and these technologies are quite useful, because you actually can, if you do the, and I'll just, if you do the surveillance, if you identify the viruses that already jumped previously, you actually can go and pre-make a lot of proteins that you may be targeting. And you don't know which one will work well, because as it, for instance, turned out with COVID, it's not the spike protein that makes good tests. Like in the UK, most of the spike protein targeted tests fail regulatory. It turned out to be the nucleic capsid, for whatever reason, which is not exposed, where they have to lyse the virus and release the capsid. Those tests are performing significantly better, and we don't know it till we actually build the prototypes and done the clinical trials, and probably went to the regulators. So what you need to do, you need to look at the genetic data. You have to find out the viruses that jumped previously, or like to do cross-species jumps, and pre-make a lot of these reagents, and just have them either on stock or ready to go. So you can go and build the libraries of binding domains, and from them you can build this very cheap, possibly not extremely accurate test that you can just massively roll down. And I think this is a very important learning from this pandemic, because this one tool we almost have, but unlike the Moderna vaccine, it has made it into the mass circulation because it's not a medical device. It's a public health tool, and we don't know how to manage public health tools, how to manage individual choices. If you swipe yourself in the morning and you turn positive, you're still going to work. Who is responsible for all these questions? But not having these things at all leaves us at the measures of draconian 2 months quarantines and a huge economic impact. And I think that where these technologies are very important, and they're not expensive, and it's actually not expensive to do surveillance and make all these things, and there's a lot of important science there. So I think that's, for me, was one of the important learnings of the pandemic, and that the things that we've done. And so now I just like to thank people from our South-Free team, the Shiley, Wayne, Jingling, May, and the survey who developed the system way back, and Dmitry, David, and Nevan from the US, who were kind enough to support a little lab on the fringes of a civilized world that wants to play in the COVID space, in the middle of the pandemic. And thanks you for your attention.
Thank you. Thanks for your talk. I'm quite interested in what you describe as the cell-free expression system. One of the kind of issues that you mentioned is, like I said, low yield compared to is there, like, although there's very low yields, how likely is so we can actually scale that up beyond like a like how we can use cell free extracts and then just produce a lot of protein. Is that something that you can see has potential, or is going to end up only research based?
Yeah, so that's been achieved. So there are one company, so Sutra Pharmaceuticals in the US, that uses E. coli cell-free system for production of biopharmaceuticals, specifically for drug antibody conjugates, because they can use various chemistry or insert various chemistry as part of the translation process, so they can conjugate drugs to antibody fragments. Having said that, if you think then a number of companies that use in vivo expression for making all kinds of stuff, and only one successful company in vitro world that tells us something about the technology. So there, I think there are like 2 to 3 successful companies started on cell-free technology. There are thousands of biotech companies using other platforms, so that probably gives you this sense, what the limitations are. So the Sutra runs their cell-free at 100-liter scale, and they have a gram per liter yield. So they can make high value pharmaceutical products with it. But they are the only one who can do it.
Kirill, thanks for that. The genomic signature work, it's not an area that I'm familiar with. It just looks so incredibly powerful. And it comes back to your question about preparedness and thinking about these tools that we just can do, whether it's preemptive testing, preemptive vaccine development, preemptive drug development. How, as having gone through the difficulty of trying to explain the criticality of this and, raise money and think about it, are scientists thinking about this the wrong way? I keep thinking about it, when we talk about climate change, we talk about pandemics. How do you think we change the conversation so that people can't avoid it? Is there a way? I'm worried this experience won't get us there, but that data is so incredibly powerful to show us what we can predict, and even if we don't know it yet, right?
Yeah, look, I'm really crap at convincing, even my lab members to do something. So, I'm not going to forget the members of general public. So, I don't really know, because yes, it's like, do you see that? It came 3 times. I'm just stupid, just keep on sequencing. We're $20 million for just sequencing every bat virus and every human genome, and doing these sweeps. Some of it probably will come through the system. Obviously, now NIH will be funding this work. But whether it actually will culminate in proper surveillance, I don't know. Again, what is the business model? How do you monetize, pandemic that didn't happen? Yeah, because it's a terrible thing, how you build for crisis that never happen. Once it happened, then there is a business model. But prevention, is such a difficult thing.
So Kirill, you said your library experience rack-time analysis was not very good, but you still picked up 400 interaction candidates. Could you speculate on what might be missing from that library? How not very good are they?
Well, meaning not very good, we got them late. The quality was good eventually, just in terms of, if we think again in terms of real pandemic, because when we started, we didn't know that the vaccine would be coming. So that could have theoretically became a feeder pipeline for drug development if vaccines fell flat. And we said, we will have it all done by August. And, we're still crunching for it. We've done a lot. And that was because of the majority of the problems were in DNA. Now, there isn't one important aspect, which indirectly related to your question, that this data was extracted from single transfection. So the transfected a single frame in the cell and moving what binds to. In real world, nothing works as a single protein, particularly not with viruses. In reality, most of those open ring frame form complexes with each other and with mammalian complexes proteins, and the network. You probably can say the net is probably like 3 to 4 times more complex. And that it doesn't even start. Well, you can kind of start with most obvious and the strongest interaction. The biology is actually probably a lot more complex. We have now having that technology, you can start unwinding it because once you know how intraviral interactions work, we can use those some complexes to probe cells. But it's a bigger undertaking.
And I wanted to see what the the one that we're using at the moment has not DGA approved for over a year, but it's massive in Australia. So when we read a report, that what you say this day has not been validated. And what is really the situation in relation to the reports at the one stage, the antigen testing carries on, but they did.
Yeah.
What is those? Because I was
yeah. Well, look, I think what happened in Australia, we just very quickly, it was clear they had effective lockdowns. Australia is an amazing country, a very rich country where government would pay people to stay home and drink beer for a month, and everybody gladly did it. The only place where they could get infected was Bunnings. And then they ramped up testing because again, the public health. Well, again, I think it's a big decision. Like the US only now started rolling out those tests because like we cannot keep up with pandemic. We need to do something. Australia's like, well, no, right? We don't need all this uncertainty. We just go put everybody for Queensland Health. Then we control the numbers. We know exactly what's happening. We can do all the tracing. It's a very top-down approach, which, like in China, was very effective. So that was, of course, coronavirus in Australia is really vanilla scenario. Could be different viruses, different activity, different outcomes, and it all went wild. And then being able to test would be extremely important with nuclear in the body. In other places, these methods were partially deployed, but they never were blanket deployed and deployed consistently. Like university campuses do it, and it's well demonstrated that that really keeps the disease at bay. Even if your accuracy is like 80% every day or every few days, you know where you are in terms of epidemiology. So, and again, I would not criticize the Australian public health system for doing good. They managed the crisis using existing technologies. Right. That one was managed. But we should be able to deploy the second and third line of defense if the first one doesn't hold, because if we would have, we wouldn't have plan B.
Hi, that was really interesting, thank you. I was wondering about the archaeology aspect a little bit. How do you actually figure out that X many generations ago, a certain population was infected with coronavirus pandemic? And once you have that number, how do you figure out that target genes from there?
Well, so the way it's done, you figure out the target genes based on your proteomic data of your modern virus interaction with the cell. So you basically say, well, those genes from virus interactions with those hosts, let's see what happened with those genes in evolution. That virus is long gone. But there's a certain rate of mutation which is fairly constant. So you can look at the genes and say, most of the genes mutating with certain continuous rates, unless they are under selective pressure. Unless you're either crawling out of the ocean to grow legs, that the like genes start mutating, or under type of viruses, that system is creating new tools. And so you basically can calculate from the accumulation mutations and how quickly those changes reverse, once this pandemic got down, when it was, because the approximate rate. Again, this is not a generation 935. It's 900, 950. But swing signatures in the field were actually quite significant. There's a whole science, and I don't understand how people look at those genomic sweeps to see how evolution played out. And it's not specific to you can do the same on tuberculosis or HIV, other diseases. So it's basically whatever's under pressure will mutate, and you can tell when it started mutating and when it stopped.
Thanks very much, Kirill, for the talk, and I'd like to thank both of our speakers for was there one more question? No. Okay. I'd like to thank both of our speakers for giving two fantastic talks and sharing your time with us today. Thanks a lot.
Thank you.
So we just have a few more items to go through. If Jenny is around, I'd like to invite her up to come and talk about New England bylaws.
So I'm Jenny from NP. I think most of you here know me or know the products that we have. And just really quickly wanted to let you know about a couple of programs that we have that might be useful. So that's right. So we have a new student starter pack. So if you are a new PhD student, honors student, or master student, or if you have one of those students in the lab, we can provide basically a bunch of freebies and technical references. So get in contact with us. Also, if you are starting a new lab or you know someone who is starting a new lab, please let us know. We can offer a pretty hefty discount for an initial order. That's pretty much it for me. Yeah.
Thanks, Jenny. Put my slides up.
All right.
Technical difficulties.
Yeah, sorry about that. Yeah.
All right.
Firstly, thanks, Trent and Kirill, for the talks, and thanks to SBA Brisbane. I was reflecting on my career as well, installing the first single-use bioreactor at CSL back in 2007. 50 litres, so I felt like I played some part in our pandemic preparedness. And then we also installed all of the RNA extraction robots to do the COVID testing around Australia over the last 6 months. So Decode Science and my previous company, in vitro, played some small part in the science that was on display tonight, which is great. I've got 2 slides about Twist. The first one, I just wanted to let you guys know that you can put custom vectors or onboard your vectors into Twist.
Just wanted to make sure that everybody here is aware of that. And secondly, and I didn't realise this was going to tie in so well with Kirill's talk, but friends don't let friends clone. So let Twist do your cloning for you. We actually have a rebate scheme where that you guys are entitled to. So we can do the cloning for you at Twist, and we can do that for free. So if you're interested in that, send me an email and we can get you onto that rebate scheme. Lastly, if you look under your pillow, there's a $500 Twist voucher somewhere there, the closest pillow. And if, Jenny, if you get it, it's not yours. Has anyone found it? Maybe Alex needs to give some direction. Where found it? Where is it? Hey, congratulations. And I just got some Twist socks and wool bones also. All right, that's me done. Thanks, guys.
Thanks so much. I guess that's it for our seminars today. There's just one last item of housekeeping that I want to bring up. Alex, who's been instrumental in starting up SBA Brisbane here in Brisbane for us, will be leaving us next month, and it'll now be James that'll be heading up the note. But I'd like to take a moment and acknowledge Alex's contributions to SBA Brisbane, who, I mean, without him, I don't think any of us would be here today having these great talks. So we can give him a round of applause. That'd be great. And with that, I'd like to thank everyone for coming out tonight, and we'll see you again next time.